Hello students and welcome to Home Learning Biology. My name is Mr. Twinzy, Mr. C. I'm inviting all of you from 5s, from 4s out there, from 1, 2s and 3s to come for this lesson which is on the economic importance of microorganisms. That is how microorganisms are useful. Our starting point will be to redefine what a microorganism is. So let's start from the beginning now and look at the economic importance of the microorganisms as we have introduced that in this lesson. Economic importance of microorganisms. I have said microorganisms are the very, very small organisms which you cannot see with your eyes. That is why they're called micro. You need instruments like a microscope to observe them. The microorganism is placed, you have a slide on a microscope, then you observe, then you'll find it there magnified, that is made larger. Economic importance, that is how useful these microorganisms are. We looked at the kingdoms in my introduction: the bacteria, algae, fungi, plants, and animals. So it's important for you to know these these 5 kingdoms and know which organisms, specific organisms, falls into which category, which kingdom. Let's look at my introduction. Thus, some microorganisms are harmful while others are useful. Harmful microorganisms, useful microorganisms. Talk about bacteria. We have bacteria that will cause illnesses, that will cause illnesses, and some that are useful. For instance, dental decay. We talked about dental decay earlier. That is the rotting of the teeth. We said it's as a result of a bacteria, the bacteria in the mouth, feeding on the sugars from the foods that we eat, the sugars which remain in the teeth if you do not brush or rinse your teeth properly after a meal. The bacteria will feed on the sugars, thereby produce acids which will wear away the teeth, resulting in the situation dental caries, as the rotting of the teeth. That's the bad side of the bacteria. That's the harmful side of the bacteria, causing of dental caries in the mouth. Dental caries in the mouth. Now, what is the usefulness? In what way are bacteria useful? How is bacteria useful? Fermentation process. In the making of cheese, that's where, bacteria is added into the milk, then it will turn the milk into the cheese. And other products, as we'll see, in this particular lesson. Okay? That is fermentation, or right here, ferment. Fem for fermentation. That's the usefulness of the bacteria. Fungi, likewise, as well. We have the good side and the bad side. But like I said, in today's lesson, we're looking at the good side of these microorganisms. Point number 2. Some bacteria, yeast, and fungi are used in food industries. They're used in food industries. Food industries like what? I want you to think of at least 2 food industries in the country. Yes, Parmalat. Parmalat is one of them, where there is milk processing, milk and juice processing. Southern breweries, okay. They make the making of food drinks, the bottling of soft drinks there. Southern bakeries, bread making. There are so many of them, okay. SUB, there are so many of them out there. These are the food industries. That is where then these microorganisms are employed, are used. The bacteria, the fungi. I'm talking about here the good strains, the helpful bacteria. That's where you'll find them in sachets and packets, and they're added in the raw material. Raw material being the starting point, the starting material, where you add these microorganisms. Then they will turn the raw material into the product that you want, into the bread that you want, into the sour milk that you want, that is wanted, into the cheese that is wanted. So that's what happens there. So it's important to know that reaction in an arrow form. Bacteria plus raw material forms yogurt. Bacteria plus milk forms cheese. They know the conditions. At this temperature, at this pH level, as we said earlier, that the action of these microorganisms need those external environments to be regulated, to be controlled. If there are extremes, then the bacteria will not be able to function properly, or they may have to get deformed. So that's what I'm talking about here. Then the next point, the yogurts, cheese, bread, microproteins. These are some of the products of these microorganisms we're talking about. I want us a short exercise to list, by means of an arrow, show the microorganisms responsible in the making of cheese, even though we're still going to look at that in more details. Cheese. Which microorganism is responsible in the making of cheese? Or rather, in the making of yogurts? Let's start with yogurts. A bacteria. A certain strain of bacteria, yes. Certain strain of bacteria, that's very good. Cheese. Bacteria again, that's good. Bacteria again is used in the making of cheese. Bread. I can see you saying yeast. Yes. Yeast in the making of bread is so yeast is added in the dough, which will turn the dough and swell it up to produce the bread. The yeast will be responsible for the evolution of carbon dioxide that will swell the dough during the heating process, and then the bread is produced in that process. Microprotein. Fungi, that's very good. Fungi is applicable here. So you need to know where these microorganisms are used, relative to the food products. Bacteria is for yogurts. Useful bacteria, not the bacteria that will cause illness, not the bacteria that will cause diseases. Useful bacteria in the making of cheese, yeast in the making of bread, fungi in the making of microprotein. So that you need to know Form 5s and Form 4s as you prepare for your examination, and also general knowledge as you prepare for your life careers. I want us to look at this one here, which is the image of a yogurt. Yogurt image. I've already introduced the making of yogurt, that it's made from a bacteria, a certain useful strain or type of bacteria. This one is a strawberry yogurt, as you can see here, which means the strawberries have been added here. Why do you think strawberries are added here? Added here. First and foremost, what's the raw material in the making of yogurt? That is milk. Milk, that's good. Milk is added. Milk is the raw material. Then the bacteria is added into the milk. A certain strain, type of bacteria is added into the milk, which will coagulate, turn it into the yogurt. If you add a different strain, it might turn the milk into something you do not want, like a cheese, But here you want yogurt. So the kind of bacteria that must be added here so that yogurt is produced. And also, their conditions must be regulated. The controls must be set appropriately so that you get the type of product that you want, in this case, which is yogurt. Now, talking about biophysics. Putting a little physics into this process here, the density of the yogurt. It is made such that it meets a particular level, particular standard. You wouldn't like to drink yogurt like you are drinking soft drink. So it means the yogurt must be made to be at a certain density. And that's a bit of physics. Why so? The strawberry is added there if the density is very low. If it's more watery, you'll find all the content, the ingredients, those stuff which you've added, going to the bottom. But then when you feed with a spoon, you would like to, each time you scoop, you would like to have a bit of strawberry into that, not have everything at the bottom. So that is that's the physics part of the processing of this, a tasty product, which is yogurt. Strawberries are added, yes. Flavored yogurts, flavor. You add flavor there. Different yogurts taste differently, so there must be a flavor added. Strawberry flavor, orange flavor, lemon flavor, all those things are added in here so that it appeals to the end customer, to the buyer out there. So all these are substances that are added there. You must know what's the role of these flavorants and all that. Now I want us to look at the next slide, which is about more information on how the yogurt is actually made in the industry. We're saying here the making of yogurt. Look at that. Fermentation of milk by bacteria. I've just said that the microorganism added in the making of yogurt is the bacteria, and the process is fermentation. Fermentation, note that, that's fermentation. The milk is fermented by the bacteria that is added there, by the helpful bacteria, not just any bacteria, as you will see here. A kind of bacteria is added into the milk to produce the yogurt. Line number 2, microorganisms and conditions are controlled. Conditions are controlled, and the microorganisms are controlled. That one is very important. Think of the making of sour porridge at home. Maybe, let's say, sour milk. The sour milk that is made at home is different from the sour milk made in the food industries. In what way? In the food industries, the bacterial strains, the type of bacteria, there is control on that. There is monitoring. Not every bacteria is allowed into the milk to ferment the milk so that you get the sour milk. So the taste will always be uniform. The taste of the sour milk will always be the same wherever, or rather, day in, day out. Whether it's a Monday, it's month end, it's November, the taste will always be the same because they know we have to add this amount of bacteria and only that type of bacteria into the milk at this temperature to get the product that should always taste the same. But at home, we get the milk from the cow, place it there, leave it open. Day 1, we come and taste. Oh, it's still turning, it's still working, it's still more milky, it still tastes more like milk. Day 3, we taste again, so it's turning sour. There you are. Where do the bacteria come from? From the bacteria that will change the milk, our raw material. So all forms of bacteria are exposed to that milk. So that is why then it will taste different from the milk that you buy from the shops in the cartoons, Yeah, so that's what we're talking about here. So line number 2 is saying microorganisms and conditions are controlled. That is the controlling aspect of it, ensuring that only that type of bacteria is added into the milk all the time so that we get that kind of taste with regards to the yogurt. Whether buy it in South Africa or Swaziland, it should be like that. To assure the wanted product, that's the assurance. Yes, in industries that is called quality assurance. Quality assurance, the quality should just be the same, irrespective of where you buy the yogurt. If you buy a strawberry yogurt, it must just taste the same. If you buy a strawberry yogurt made by this particular company, it must taste the same year-round, irrespective of where you are. Line number 3. 2 kinds of bacteria are added in the milk. 2 kinds of bacteria are added in the milk. Note that, 2 kinds, not just all the bacteria, 2 kinds. The helpful bacteria which will give that strain or that taste of the yogurt. On a commercial scale, the fat and protein content of the milk is adjusted and the milk is homogenized. The fat and protein content is adjusted on a commercial scale. Commercial scale as opposed to the homemade products, the homemade sour milk, the homemade cheese, the homemade yogurt. Let's talk about the homemade yogurt. Commercial scale, that is for sale purposes, for sale, products for sale. The fat and protein content are adjusted. You'll want to have the fat level in the yogurt minimal. I mean, you go to the shop, you get the yogurt, the can, the bottle of yogurt. Then you have a choice. No fat. Oh, I like that one, no fat. Or if you want the fat, cream. There's cream here. A lot of cream, a lot of fat. Then you decide. So we're saying here, that is adjusted so that a customer who wants a yogurt with no fat will take that one without fat. So that's the control aspect of it. Quality assurance, quality controls of that. And the milk is homogenized. To homogenize is mixing. Homogenize, mixing, so that it's the fat content in the container is the same at the lower end, at the top, and at the sides. Remember the stirrer we talked about earlier? So it's for homogenizing. It's for homogenizing. So mixing up so that the level of what we're talking about here is the same. The concentration of what we're talking about is the same, irrespective of the part, in the inside of the container. This mechanical process breaks the fat droplets and prevents them from separating out. That's the breaking of the fats in there and preventing from separating out. In the industries, there are special tools for doing that, like the stirrers, which break the fat and homogenize. So it's a very, very interesting field on that regard. So this mechanical process breaks down the fat droplets and prevent them from separating out. Separating out, that is, when you drink the yogurt, we're talking about yogurt here. As you drink the yogurt, you wouldn't like to have, at the top of the yogurt, it's more watery. Then at the bottom, it's thick. That will be bad. That's bad for the quality of the yogurt. So quality controls there may have been compromised as the yogurt was being processed. So to counter that, to prevent that from happening, then the mechanical process of stirring, homogenizing is employed. The milk is then pasteurized. Pasteurization is about what? Removing pathogenic organisms, removing disease-causing organisms so that it's healthy. You don't want to buy a yogurt, you don't want to buy a product which will cause you illness thereafter. No. So we talk about pasteurization here as that element. Pasteurization destroys the pathogenic bacteria, like I've just said, destroys the pathogenic bacteria and other microorganisms that might cause unwanted taste. In the food industries, in particular milk production, or rather the making of cheese and the making of yogurts, they are opportunistic microorganisms of bacteria which may find their way into the product and then spoil the product. That is bacterial spoilage of the yogurt, or yogurt spoilage due to the bacteria. So they try to separate those opportunistic microorganisms from coming in. For the product will therefore become different. What do you mean here? You buy the yogurt from the same, the same brand of yogurt, same manufacturer at Hlangano. You find they taste different from the one you bought at Pigs Peak, because one bought down south was made, say, in May, and the one bought up north August. So somewhere along the line, there must have been an entrance of the opportunistic bacteria which change the flavor. This bacteria will come with associated with the flavor of the end product. So that's breaking or compromising the quality. So it means the quality assurance, the aspects must have been not at the right levels. So that's what I'm talking about. At this level, we're just saying a particular strain of bacteria must be used. It must be ensured that it's only that in order to get the taste of the product that we're familiar with. Pasteurized milk is then fermented by adding starter culture. We call this bacteria starter culture. The bacteria that is useful, that is added there, is called the starter culture. That's the terminology used in the food industries, which you'll get to know about it when you do your second year in the food processing course at tertiary institution, Form 5s. Streptococcus, Thermophilus, and Lactobacillus bacteria are used. So it's not necessary for you to know those strains, these types of bacteria, but what you need to know is that only certain types of bacteria are used in the making of yogurt, not just any. So these two are used in the making of bacteria. They act. Look at what these bacteria do. They act on the milk lactose. They act on the milk lactose. Lactose is the sugar. They act on the milk sugar, converting it to lactic acid. Converting it to lactic acid. The yogurt will taste a little bit bitter because of the acidic nature. Remember, from a chemistry, acids taste bitter. Not to say you should go there and taste acid. No, never do that. Otherwise, you'll have no tongue thereafter. So they taste bitter, lactic acid, which in turn coagulates the milk protein casein. Coagulates, that's forming the card. Thickens the milk. Thickens the milk protein to produce a thick, creamy consistent of yogurt. So what are I saying here? I'm saying you're adding this strain of bacteria, this type of bacteria into the milk. It will act on the sugar lactose of the milk, change it to lactic acid. The lactic acid, which will act on the milk protein and make it thick, a bit thicker. You know, remember milk is watery. So the lactic acid will make it a bit thick, and that is what is want. Thick in line with the thickness of a yogurt. Not in line with the making of cheese. You know, in the making of cheese, it must be very thick and solid. So we are looking at here the yogurt. So that's what it's all about. So in the food industries, the technicians, the food technologists are always looking at such matters. So I hope that will generate interest in you as you work around what to do when you finish high school, when you finish your Form 5. Next line, fermentation works at 46 degrees Celsius. Yogurt then cooled to 5 degrees Celsius to stop the bacterial processes. The important aspect here is the control. That's the control aspect of what we have been talking about. So I'll write here controlling the temperatures, 46 degrees Celsius, and yes, controlling. So all about control. As the temperatures, you know, bacteria, some bacteria are denatured at very high temperatures. Some are denatured at very low temperatures. There must be optimal temperatures for their function. So if you know now, no, the yogurt is now at the right taste. The yogurt is now at the creamy level that is wanted. You may decide to lower the temperature. So the bacteria which you added there will then die because the conditions will no longer be conducive with regards to the temperature. So if the bacteria die, there will no longer be fermentation. So the bacteria will no longer produce lactic acid, making it more sour, more bitter, or rather deviating from the expectations, from the standard taste of the yogurt. So these are the controls. You either lower it or raise the temperatures so that the bacterial aging stops. Then you start getting your yogurt, packing it, taking it for sales in the shops. Lactic acid gives the yogurt a slightly sour taste. Like I said, acids taste sour. Not to say as a warning though, you must taste the acids in the school laboratory. No, that is never done. You can only do that if you want to lose your tongue. So they have a slightly sour taste that accounts for the taste of the yogurt, the lactic acid. Some important points to note with regard to what we are talking about here, and pasteurization. These notes here are very critical for your examination purposes now as Form 5. Milk is sterilized by heating to high temperatures. Sterilization by heating to high temperatures. Go to a milk processing plant, juice processing plant, or industry. You'll find a series of pipes, a lot of pipes from point A to point B across this very large room. The milk, which is the raw material, is placed at one end and is channeled, moved along the pipes to the other end where it is collected as the end product. Along these pipes, there will be a heating area where a lot of heat will be applied to the pipes. When the heat is applied, the bacteria which are accidentally inside the milk, in the pipes, will be killed. But there is control such that the nutritional value of the milk is not destroyed. That is part and parcel of pasteurization. Pasteurization, it comes in many ways, of course. If you look at a carton of milk, you'll find pasteurized milk, ultra treatment. So these are the elements I'm talking about that kill the bacteria inside, so that as you buy from the shop, you are guaranteed safety. The health standard has been monitored during the processing line. The second line here explains all that. This kills any other bacteria already in the milk which might spoil the flavor of the yogurt. You get the milk from the cow out there, and during the process of milking, the milk is exposed to the bacteria. It's brought to the process line with the bacteria. Then, during the sterilization, the pasteurization, the ultra heat treatment process, the bacteria which accidentally got to the milk is destroyed, it's killed. So that keeps everything in good order. The lactic acid gives the yogurt a pleasantly sharp flavor. That's the lactic acid, pleasantly sharp flavor. And the lactic acid concentration, like I said, is controlled relative to the kind of yogurt that is wanted. Flavoring such as sweeteners or fruit can be added. We saw earlier in our image of the yogurt that strawberries was added into our yogurt. So that is a strawberry flavored yogurt. And is packed into sterilized containers for sale. That is the end of the line, then packed for sale. So as you buy the product in the shop shelf, you see, oh, that's strawberry, and you buy that, you are always assured that it's from this reputable company, so it was pasteurized, it was sterilized, so drinking it, eating it, feeding on it will be healthy for me and for us all. So students, I want us to take a short minutes break, and from here, after the break, we shall look at the making, the processing of cheese, and all the nitty-gritties involved there. So stay with us. I can't sit. I can't sit. Hello students, and welcome back from a short break. I hope you are energetic now as we are going to look at juice, the juice making, and all the nitty-gritties involved there. Juice making. The juice is what is found in the fruit, and it contains the nutrients that the body needs. The traditional way of making juice is take the fruit, squeeze, then there you are, you drink it, and you have all the nutrients there. In the food industries, because we're talking about these applications in the food industries, they have mechanical or very sophisticated instruments or machinery that will do the squeezing within a short space of time, other than having us as people do the cutting, squeezing, which will take longer. So that is what we're talking about. If you look at this slide here, there is the orange juice in this glass and the orange next to it. Juice making, you cut it, then you squeeze. There you are. You are sure of getting a 100% juice by so doing no water added. That's the most healthy juice. The juice comes in many forms with regards to how the customer wants the juice. It may be more liquid, clear, cloudy, or there is an element of the treatment of the juice. For a cloudy juice, all the parts of the orange, as you can see here, all this part here, part of the orange, will then be sieved off from the juice. Will then be sieved off from the juice if you want to make a clear juice. But for it to be a bit cloudy, that will be incorporated, will be retained in the juice. So that is juice making. Let's look at the processes, what exactly is relevant to our syllabus as we do, as we study the juice making. So we are saying here, students, juice making. When juice is squeezed out of apples to make a drink, the enzyme pectinase is added. Enzyme pectinase is added. Enzyme, what is an enzyme? Biological catalyst that speeds up the rate of a reaction without taking part in the reaction. So when the juice is squeezed out of the apple to make drink, enzyme pectinase is added. So this one is with reference to apple juice, not orange juice. But always know that enzymes are always responsible. That is why for some fruits, it's advisable to eat the fruit wholly as is, if that is possible. Like fruits like apples, we eat everything. So the inner side of the fruit contains those enzymes. Then you are home and dry that you will benefit from them. In instances where you peel the cover, then the peels may go the enzymes, then will get less. So that's what I'm talking about. So when juice is squeezed out of the apples to make a drink, an enzyme pectinase is added. What's the role of this enzyme pectinase? Pectinase breaks down the substance that hold the cell walls of the apple cells together. Breaks down the substances that hold the cell walls of the apple cells together. Apple cells are held together. Apple cells are held together. The cells are held together. Cells are always in contact in plants, in animals. One cell in contact with another. Cell wall in contact with another cell wall. So the pectinase will break the cell walls. If you break the cell wall, so you are creating space. You're creating some kind of porous nature, some kind of holes for the juice to be able to squeeze out easily as you compress them. So the pectinase does that. Instead of doing the traditional way, puncturing the apples with a sharp object, puncturing like that, then squeezing, cutting, adding pectinase will remove the cell walls so that the juice is extracted more easily. So that is an industrial operation. So this makes, like point number 3 says, this makes it easier to squeeze out more juice once the pectinase enzyme has been added. Technology will bring about all this, biotechnology. That's biotechnology, the use of biological microorganisms in the food processing. But we are seeing, as we're seeing here, the use of the enzyme pectinase which will enable the squeezing of the juices to be much easier and faster. And so the process will roll out on the shelf. Then, last point, it turns the apple juice from cloudy nature to a preferably clear liquid. Remember I said when I showed you the image of the juice that you have an option of taking a juice that is more cloudy or one that is clear. How do you do that? Mechanically, you may use a sieve, a strain, a sieve, a filter, and filter all the particles that you do not want from the cloudy juice so that from the receiving end we have a clear juice. But this one also, from the mechanical aspect in the industries, you add the enzyme pectinase, which will, as we have said, turn the apple juice from its cloudy nature into a clear liquid. So that's a very, very important point. You want to turn it from cloudy to liquid nature. Then you can always label on the bottle, on the container that this is cloudy juice, this is a clear liquid juice. Then the customer will always have a choice on what to buy. So these are all the elements you find in the food industry during the food processing. Let us get to milk now and look at how milk is. All familiar with milk? That is it. White as is from this slide. That is the milk. And then we look at milk processing. We said earlier, just to recap, we said that for some people who drink milk, they may have some illnesses thereafter. They are allergic to raw milk as is. So the milk has to be processed a bit for them to be okay. That also varies, depends with people. Some people are okay, they just drink the milk and they still feel okay. So in this lesson, we're looking at what causes that little discomfort when you drink milk as is, as a person. And how is that discomfort controlled? So let's look at that aspect of milk processing, Form 4s and Form 5s. Point number 1. Milk enzyme, or rather enzyme lactase, breaks down sugar lactose in milk to glucose and galactose. Note this equation. Note this equation here, this reaction. The enzyme lactase break lactose. So we're starting with lactose, and we're saying arrow. It forms glucose plus galactose. And the enzyme here is lactase. And I'll give it a different color for the name of the enzyme because it's lactase. There you are. Enzyme lactase works on the lactose. The lactose is the sugar found in milk. The sugar in milk. Lactase is the enzyme. Remember, ASE for the enzymes. Enzymes always end with ASE. Lactase, amylase, lipase, lactase, protease, ACE for enzyme. So the enzyme lactase acts on the lactose to produce glucose and galactose. Let's look at the point again. Enzyme lactase breaks down sugar lactose in milk to glucose and galactose. Why so? Some people cannot digest lactose and it makes them ill. Digestion. The milk is food. Food is taken in through the mouth. There has to be digestion of the milk down the alimentary canal, you know. Stomach, mouth, stomach, all the way down. Duodenum, there has to be digestion. So I'm saying here, some people cannot digest the milk due to the lactose in there. And as they try to digest that, it makes them ill. So if it makes them ill, and milk is a very important food item, for it contains so many nutrients, one is to come with a solution. There must be a solution to that because they still need the milk. What's the solution? Lactase digests the lactose. So lactase digests the lactose. Lactase digests the lactose, and so what? Let's see. The resultant lactose-reduced milk is safe for people to drink. Oh, so that means in the milk, lactase is added so that the lactose is reduced, is removed, is changed to glucose and galactose. So the milk will now have glucose and galactose. So the people who are allergic to raw milk will drink milk with glucose and galactose, which is healthy, which is safe. I have safe here, which is then safe for people to drink. So that means that there'll always be milk for all of us out there. For those of us who are allergic to milk raw as is, we'll drink the milk that has the lactase enzyme added. For the lactase will change the lactose, which causes us ill, which causes some people ill. Then we change to glucose and galactose, the glucose and galactose, which is okay with them. So whenever you think of milk processing, raw as is, at that elementary level, at that basic level, always think of the enzyme lactase acting on the lactose found in milk and changing it to glucose and galactose, which is healthy for everyone still, which won't cause some people allergic to milk ill. That is milk students. Know that point, and always figure it out whenever you see an image of milk. Next, I want us to look at cheese. I want us to look at cheese. There's so many kinds of cheese out there. You can name them. There are so many varieties of cheese. It all depends on what strain of bacteria is added to the milk to produce the cheese. The colorants added in there, the temperatures at which the milk is processed, or in general, the conditions, the environmental conditions under which the bacteria work, including the pH, including the exposure of the milk to the bacteria, all that are important elements in the production of the final product, that is the cheese that we want, including the taste, of course, including how soft, how hard the cheese is. Remember, when we talk about yogurt, we said exposure of the milk to the bacteria will produce lactic acid. The lactic acid will change the milk, will harden the milk into the yogurt. So that means more exposure of the milk to the bacteria will have the milk curdling more to form the yogurt. So what does that imply? It implies that even more exposure of the milk to that strain of more exposure of the milk to a particular strain of bacteria will make the cheese to harden and harden and harden until you get the kind of cheese that you want. Less exposure will give those soft cheeses. So what I'm saying here is it all boils to the word control. The word control all boils down to the word control. We control the conditions. The conditions must be controlled to get the kind of product that you want. Bread, cheese, bread, yogurt, you name it, the conditions must be controlled. Pay particular attention to this, as you may find yourself working in a food industry where you'll be processing cheese later on in your life. Not very far from, after finishing school, after finishing tertiary education. Point number one, a variety of bacterial cultures. Remember, we said we call these cultures bacterial cultures. That's the terminology used in food industry. A variety of bacterial cultures may be used to ferment milk. Fermentation. You must always think about an acidic product being produced. Fermentation, sourness product, a sour product, an acidic product, fermentation, with some carbon dioxide, with some ethanol, with some acidic lactic acid. Always think about that. But once again, it depends on what fermentation process you are talking about. For instance, some fermentation will produce ethanol as a product, some carbon dioxide, lactic acid, it all depends on what is being of interest. What is of interest there. But I want us to look at this. A bacterial culture may be used to ferment the milk depending on the type of cheese that is produced. Yes, the type. So the keyword here is type. Keyword is type. Streptococcus and lactobacillus work at 40 degrees Celsius. Yeah. Now, if you take a different strain of bacteria, you may find that it works at 35, at 30, then you won't get the product you want. So for Streptococcus and lactobacillus, if you want to get a particular kind of cheese, then you must control the temperature to be 40, as specified here. But for examination purposes, never mind the temperature, never specify the temperature. But you must know that, always say that controlled temperature conditions. A mixture of enzymes called rennet is added. Cheese making, mixture of enzyme, rennet. Know that rennet is a mixture of enzyme. The examiner may give you the term rennet. So you must know that, that's a mixture of enzymes. Rennet contains enzymes chymosin, which coagulates means coagulation, thickening, which coagulates the milk casein, that's the milk protein, and forms a semi-solid curd, semi-solid. So we talk about milk, watery, then semi-solid curd. From milk to the semi-solid curd, so we're in the process of thickening the milk to form the cheese now. Chymosin enzyme is mainly produced from genetically engineered yeast. There you are. The enzyme chymosin, from where is it produced? Genetically engineered yeast cells. Chymosin is an enzyme from yeast cells. Enzymes from yeast cells, which carries the chymosin gene. Okay, you know the chymosin terminology, chymosin enzyme. Chymosin is also obtained from calves' stomachs, from the stomachs of calves. The same enzyme chymosin is found in the stomach of calves. 1, 2, genetically engineered yeast cells. That's where you can get the enzyme of interest. Yeah, use chymosin. The liquid whey is drained from the curds. Curds, remember, are the solids now. The milk has turned into a solid kind of substance. And as it turns into a solid, there will always be that liquid part. Have you seen the making of sour milk at home? Yes. Just at the maturing stage, you always find the liquid part at the top, then the solid part at the bottom. So, in the making of cheese, that liquid part is whey. So in the formation of the thickening, because even the traditionally made sour milk is thick, it's different from the milk. So there'll be that liquid part. Even here, in the food industries, when they're making of cheese, there'll be that liquid part, the whey, then the solid. The solids are separated from the milk, from the liquid components. That's where the whey comes from. So the liquid is drained from the curds, which are partly dried and compressed. To be used for other purposes, of course. To be used for other purposes. So there's nothing to throw away here. In the food industries, there's nothing to throw away. So there'll always be good use of whatever product is there. The degree of drying and ripening depends on the variety of the cheese. On the variety, there you are, variety. Camembert cheese, this cheese, that cheese. You may go to any shop, especially supermarkets, you'll find all the different kinds of cheeses there. And how they come about is a function of the degree of drying. The degree of drying will determine the kind of cheese that you want, that is wanted out there. The next point, bacterial enzymes act on the proteins and fats in the curd, partly digesting them into amino acids and fatty acids and giving the flavor. My point here, my interest here is the flavor, the aroma of the cheese. Flavor, that is how it tastes. Aroma, how it smells. Aroma, how it smells. So that is all that is a function of the bacterial enzyme. It's a function of the bacteria that was added there. So it will determine the taste and how it smells, how the cheese smells. Then when you come as a customer, you say, that's the cheese I want to buy it because I've seen it. And on top of that, you add what? The colorant. You want a yellow cheese, a white cheese, so that also is an important aspect for marketing purposes. With some blue cheese varieties, penicillin spores are added. Like I was saying, the colorant, so that is a function of the colorant now as we talk about the blue nature of the cheese. That's a colorant. That's a colorant aspect. It's blue, so it's the color. You add penicillin, then there you are. You get the kind of cheese color that you want. Fermentation. So it's all an aspect of fermentation. So students, that is the making of cheese in the food industry. We do expect you to understand this in order to pass examination and generate interest in working in food industries that manufacture cheese. And better still, own a business, a process line in the future that will process cheese so that the economy is uplifted for us all in the country. So next, I want us to look at the alcoholic beverages, beer. I show you this person here, this young man here, with all the alcohol in his head. As you can see here, we'll just take it as beer. Beer. I won't bother on the name, but we'll just say that's beer with a certain level of alcohol. So how is it made? It's also a fermentation product. This is also a fermentation product. It's made in the food industries, and as you'll see, the raw materials like barley, where microorganisms are added into the barley. Then there'll be fermentation. There'll be product at the end, which is the beer, which is the alcoholic product, beer. So yes, it is an alcoholic beverage, and yes, it is employed. It is processed. It is a processed product. It's a product from the processing of raw material using microorganisms. Remember our topic here? Our lesson is about economic use of microorganisms. As we see here, the making of beer, the making of alcoholic beverages is all a function of a raw material which has a microorganism being added to it. That is alcohol. So with regards to alcohol, we shall quickly look at that. Making of beer, we'll run through this one here. The fermentation is brought by yeast. In the making of beer, it's brought by yeast. We have seen with cheese that it's a bacteria. You've seen it yogurts that it's bacteria. Here we're saying it's yeast. Yeast is applicable here. It's made from barley. So barley is the raw material. So it's barley plus yeast forms the it's fermentation process. Then you get a beer. Barley grains are soaked in water for 2 days, 15 degrees Celsius. That's the control. That's the aspect of control. All right, CNT for control. Germination. During germination, the stomach digesting enzymes in the seeds are activated. Okay? The process is malting, and the product is a barley malt. The next line, extra enzymes from the wheat or rice are added. With regards to this, I just wanted us to concentrate on the last note in the brewing process, which carries more interest. In the early stage of fermentation, yeast respires anaerobically, or rather aerobically, oxidize the glucose, producing carbon dioxide water. So that's aerobic respiration. We talked about respiration earlier, so now we're seeing this kind of respiration here, producing carbon dioxide, water, and dividing rapidly. Anaerobic, so the two forms of respiration observed here. Anaerobic respiration proceeds, giving out alcohol and carbon dioxide. So first is aerobic respiration, then anaerobic respiration, which will produce the alcohol. Alcohol is a product of anaerobic respiration. That's the important part in this making of beer. Fermentation takes 5 to 14 days, yes. Dead yeast, proteins, hops, resins are allowed to settle out, which are clarifying agents, okay. Pasteurization. We talked about pasteurization earlier, and still we are looking at the pasteurization that is very important. Pasteurization, carbon dioxide level adjusted to give the necessary sparkle. The carbon dioxide concentration is adjusted. It's a control aspect again. That's the adjusting. It's adjusted to control, to give the sparkle the unique nature of that beer, of that alcoholic beverage. And finally, flavor depends on the multitude of the substances. All that is added in them will determine how the product, the beer, will taste like. So that is beer. But again, on a very, very important note, as school-going children and students, we are not encouraged to take alcoholic beverages, for they interfere with your studies. So as we wind up this lesson, students, I want us to once again look at what we talked about. We talked about the yogurt, and how it's manufactured and how it's processed in the school. And we further looked at the juice making and the milk and cheese. I hope now you'll be in a position to answer any question that the examiner can bring in that regard. So from me, Mr. C, I'm saying, have a good day and enjoy the rest of the programs on Swazi TV. I'll see you next time around at 4:30. Bye.